Hello, and thank you for joining us today. My name is Andrew Warmington, and I'm the manufacturing editor at Sightline. With us here are Stephen Boppart and Matthew Jones, who are going to give you an overview of accelerating speed to market through solid state and crystallization development. Stephen is Director of Business Management API at Thermo Fisher Scientific. He's been with the commercial group of Patheon since 2007, having worked with emerging and large pharmaceutical clients to take projects from early clinical through to commercialization. Prior to joining Patheon, Stephen helped to pioneer and introduce polymorph and salt screening workflows and services to the pharmaceutical industry in Europe. And Matthew Jones is Senior Manager Crystallization, also at Thermo Fisher Scientific. He has over 25 years of experience in crystallization process development and solid state chemistry, and has worked in pharmaceutical development since 2008. He joined Patheon in 2016 and has since established a solid state and crystallization process development laboratory with a group of specialists. Matthew has a PhD in physical chemistry and a higher doctorate in chemical engineering. Stephen and Matthew, over to you.
Thank you, Andrew. Hi, so before we get into the topic at hand, I'd like to give you a brief overview of the API facilities at Thermo Fisher. We have 5 sites globally, 2 in Florence, South Carolina, 1 in Cork, Ireland, 1 in Regensburg, Germany, and 1 in Linz, Austria. The early phase sites in Florence and in Regensburg, there we typically work from preclinical all the way up to phase 2, typically scale of a few hundred grams up to maybe tens of kilos. And on our commercial sites, we typically work from tens of kilos and in some cases all the way up to hundreds of metric tons. We've been doing this for over 50 years, have had extensive experience. Since 2014, we brought in 400 new projects. The bulk of them have been in preclinical in phase 1 and phase 2, but we also do many late phase and commercial products as well. Having all that experience gives us a tremendous amount of knowledge. We've done this before many times. Our project managers are very experienced. Our PR&D and AR&D chemists understand what it takes to bring a product through the various stages of development and then registration and validation. We have sourcing expertise around the globe with boots on the ground in Asia, as well as lots of people here in North America and in Europe. So anything that you need with regards to developing a process, we've done many times before. We have the experience. Some of the key capabilities that we have from a production standpoint, we have high pressure manufacturing. We do lots of work with advanced catalysis. I think currently we have 5 commercial polymer APIs, which is a bit of a niche for us, and also continuous flow chemistry. The first commercial scale flow chemistry product that we had was in 2000. That was a non-pharma product, but we scaled up our first GMP flow chemistry process in 2010 up to hundreds of metric tons, and since then have had a number of GMP processes, both at very low temperature and high temperature. We also have spray drying capabilities in our Florence, South Carolina site and also our Cork, Ireland site, and can handle low and high potent products. So next slide, please. So I'm going to talk to you a little bit about why. Why would you want to implement crystallization? I think these topics are fairly well understood by everybody, but it's good just to review them as well. First, having good crystallization processes gives you tremendous efficiency and ultimately cost in the process, or taking out costs in the process. You need to have an effective control strategy to make sure that you're achieving the desired specifications and purity. And in addition, your control strategy also talks about or impacts the number of GMP steps required. Anytime you're talking about impurities, that has an impact on your analytical development and the requirements. The form that you choose ultimately for your API can affect the stability of the product as well as the intermediates. That also can impact the bioavailability of the compound and the downstream processing, the compactability of the product, the flowability of the product. And then finally, polymorphs can be used to broaden your patent protection and even in some cases extend the patent life of the product. Next slide. So as you develop your process from tox to commercial, a lot can change. Your synthesis route may change, reagents or catalysts may be optimized, filtration might need improving, unforeseen scale-up effects might need to be addressed. And understanding the impact on the crystallization is very important for every one of these changes. It's also important for the drug product development. Changing particle size or form can impact the stability, how well the product behaves, and also compressibility, for example, in the tablets. So really having a good understanding of your API solid state properties and characteristics can ensure that the API and the drug product manufacturing sites can manufacture this in a consistent, robust, and economical manner, resulting in consistent dosing for the patient in a safe and efficient manner. Next slide, please. So, as I mentioned, when we often get a product from Medchem, we have chromatography. It's an easy way to purify material on small scale, but when you get to larger scale, it starts to have some very negative impacts. It typically is a very poor yielding process. It uses expensive solvents, expensive stationary phases, zeolites, for example, lengthy unit operations. And so we try and get away from that. In crystallization is the most common method to replace chromatography. Why is that? Well, we typically can get steps or crystallization steps that give us high yields, in many cases at or above 90%. But it's important to understand your process conditions, and Matthew will get into this a little bit more detail, but the purity of your crystallization is going to be greatly impacted by the solvents or the salts that you select. The particle size has a huge impact on the speed of filtration. Ideally, we'd like to be at 8 hours or definitely less than 1 day for a filtration step. But having the wrong shape can result in the filter bed compacting and can result in very, very lengthy filtrations, in some cases up to a week, which is just not feasible in full-scale production. One aspect which is not so commonly understood is the ability to provide a hold point or an isolated step in production. And that can be really beneficial in production planning. And here I have an example where we have 4 steps. The first stage takes 3 months, 10 batches, and because it's dilute. And then stage 2, we go into 3 batches. Stage 3, which is a conversion synthesis, also 3 batches. And then stage 4, 7 batches. And as you can see, that the timing is very different. And if we were to go from batch 1 of stage 1 directly into batch 1 of stage 2, we would be tying up the equipment in stage 2 for the same period of time that we would have stage 1. And so it can be very beneficial from a production planning, from a cost perspective, to produce all of stage 1, to isolate each individual batch as it's produced. And solid material tends to be more stable than liquid material, doesn't need as much space, it's easy to pack out. And this can give us tremendous flexibility in our production planning, and as a result, can help with the costs of the overall commercial production. Next slide, please. So here we have, it's a slide I actually took from our validation presentation where we talk about fate impurity studies, but it gives a very good depiction of what can happen to impurities as you go through the various stages. So here you have stage 1, impurity A1, for lack of a better term, that will, as you go through the chemical transformation, be it an oxidation or a coupling or whatever, that impurity can react just like your product does and give you other impurities. So here you have stage 2, you have A1 and B1, and that can then react further in a following transformation or step into additional impurities. And so it's always good practice to see, can we control the impurities as early on in the process? And how do we do that? Well, ultimately, it's best if you don't produce these impurities, and so you look at the reaction kinetics and or conditions and whatnot to minimize those impurities for sure. But in many cases, you don't have, you're not able to get down to the levels that you need to, and so you need to have a purification step. And so we look to see, can we purge these impurities? And the most common way of doing that is crystallization. Other ways you can do that is with distillation or maybe extraction. But as you see, if you have a crystallization step, you can many times purge those impurities. And as I mentioned earlier, the reaction, or sorry, the solvents that you use and the process that you use can have a huge impact on how well those impurities are purged. So next slide, please. And this is another slide I pulled from a presentation we have on designing and designating your RSMs, your registered starting materials. And we're seeing in the last few years that the regulatory authorities are pushing back and asking that you produce more steps under GMP, that you have RSMs earlier on in the process. And why is that? And ICH Q11 states that all processes have to have a set of control strategies. And if you look at the second bullet point here, control strategy, designing the manufacturing process, and yeah, as we talked in the previous slide, crystallization is one of the best options for purification. So by having purification in various stages of the process can have a really positive impact on controlling the impurities and can give you a robust process that will give you always the material that you're looking for with the right specification. So generally, the more isolation steps you have, the better the process will purge the impurities. As a result, you may be able to have fewer steps produced under GMP, and that will allow you to source your raw materials later on in the process, which are commercially available, and that can have a huge impact on the overall cost and speed, to be quite honest, of producing this in future campaigns. So next slide, please. So in summary, we're looking at really three objectives that can be achieved by crystallization. Isolation, removing solid products from a complex liquid matrix, ideally with high yields, around 90% or higher, and it can give us the form that we're looking for. We're looking to purge impurities, removal of byproducts, and also solvents to a given specification, and providing the right particle size distribution, the mean size, the particle shape, which helps us with filterability and also downstream processing. Now, this can be ideally done in one-step crystallization per isolation. That's when we try and isolate and purify the right particle in one step. Obviously, if you have one unit operation, you have fewer costs, faster turnaround. But to do this, you really need to have a very thorough understanding of the crystallization landscape. In some cases, you need to do this in two steps. The first step for isolation and purification to purge the impurities, you may need to have one form. And then in a second crystallization, you look for the form that is needed for the drug products. The advantages are if you separate a task into two, in many cases, it's easier to optimize, but you have an additional step, additional costs, yield losses, and so there's a cost implication to that as well. Next slide, please. So we talked a little bit about polymorphs earlier, but polymorph screening is really a must, both from a cost and a risk perspective, but also a regulatory requirement. When new forms appear unexpectedly, it can really have catastrophic consequences. You really don't want to have this pop up during a registration campaign or a validation campaign. This can delay the launch of the product easily by 6 months as you do a root cause investigation, develop CAPAs, and having to get back into production, so finding a new production slot. You're looking at 6 months, maybe even longer delay before you can actually file your NDA, and that has an impact on when you can actually launch the product. Worst case scenario is that a new form appears after launch. There's a very well publicized case study or case for ritonavir. This was an Abbott Laboratories product that was introduced in 1996. In about a year and a half after launch, they noticed that the product didn't perform as it expected in the drug product side. And what happened is that a previously unknown lower energy, more stable polymorph, form 2, appeared. And trying as hard as they might, they were not able to get back to form 1. So in all the laboratories and all the production facilities, form 2 absolutely took over. And that was not suitable from a bioavailability standpoint, from what they registered as a process. And so ultimately they had to withdraw this product from the market, work on reformulation, essentially live with form 2. And this ultimately cost Abbott Laboratories about $900 million. And this is $1998, so you can imagine what it might cost now. So for this reason, the regulatory authorities do require a very thorough polymorph screening by latest the end of phase 2. Most commonly, we find our clients are doing this already at pre-formulation or phase 1. Next slide, please. So the work that we do in API synthesis is not independent from that of our colleagues in the drug product side. So it's key that we have a really good understanding of the crystallization landscape. Having in-depth knowledge of the possible polymorphs and salts and solvates, co-crystals, and the relative stability is really crucial to developing an efficient crystallization process, which provides the right material for clinical and commercial supply, but also material which allows for the optimum formulation and scaling of the drug product. Next slide. In fact, probably more than any other attribute, crystallization development and characterization of the API will directly impact the formulation of the drug product and ultimately your clinical supply. So this is where the handover occurs from API to drug product. And if it's not done properly, if you have poor material characterization, you will have poor product development. Next slide. In fact, we estimate that not properly characterizing your API can result in an additional 5 months in your development timeline. This is due to having to repeat various experiments, for example, excipient compatibility or blend uniformity or compaction studies, which can also impact the need to revise your analytical methods, may impact the formulation, so you may have to do reformulation. It can pose stability challenges. For sure, you need to redesign or restart your stability studies, and you may even need to do clinical bridging studies. So we estimate that this can add easily 5 months to your development timeline and waste valuable API. Next slide. So now I'd like to hand it over to Matthew. So I hopefully gave you a brief understanding as to why you need to have a good understanding of your solid state characteristics and implement crystallization development up front. It can't be an afterthought. It really has to be part and parcel of every stage of development. And Matthew's going to delve into how we approach this work. Matthew, over to you.
Thank you very much, Stephen. The first thing I'd like to do is just give you a brief overview over our solid state capabilities at the various API sites that Stephen mentioned earlier in the day. At the Linz, Austria site, we have a group that consists of crystallization and solid state and analytical specialists. We can do a wide variety of solid state characterization. We support early phase development at our Regensburg and Florence sites, the early development site of Florence. And of course, we support crystallization process development for projects at the Linz site. In the image here, you can see the chemistry park in Linz. So this is a typical Central European chemistry park. There are many companies operating here. We are just one of about 30, and highlighted, you can see the R&D building. This is where we sit in the background. You can see the beautiful blue Danube, which doesn't look as blue as the name suggests, unfortunately. In our global network, we also have solid-state capability in Florence in South Carolina and in Cork in Ireland. Here, the focus is slightly different than in Linz. In Florence, the solid-state is focused on solid-state state analysis, in particular providing support for spray drying and for milling. And in Cork, Ireland, we have a specialist material science group. Amongst others, they also provide cross-site support for drug product development. Okay, I'd like to expand a little bit on what Stephen already touched upon regarding crystallization process development. Crystallization process development is something that accompanies the entire development timeline of your new drug product. You may not be aware, but activities actually already start in medicinal chemistry or in very early development. You will carry out some form of solid-state characterization, usually in medicinal chemistry, as a means to understanding the properties and understanding which possible formulations might be suitable for your molecule. In early development, you will start looking at crystallization as an isolation process. You will assess crystallizability, in particular in cases where you may have not been able to generate crystalline material in earlier development. Not all APIs crystallize, but very few don't. Once you've managed to crystallize, you will assess solvents, carry out a preliminary solvent selection, and then generate a crystallization process that is good enough to supply pre-formulation and toxicity studies. The real work starts in the clinical trial phases. In phase 1, you might take a closer look at your process. You might even redesign it. You start looking at physical parameters of the system that are important for process design, such as solubility and metastable zone width. You will select the process type, whether you need a simple cooling crystallization, an anti-solvent crystallization, an evaporative crystallization, or a combination of those. You will start looking at process ranges, optimizing yield, making sure that the purification works, and do initial scale-up work. But usually the amount of work is quite limited at this stage, but it picks up in the clinical trial phase 2 stage. Here you start with serious process optimization, initial quality by design activities. And this is where you might be engaging in particle engineering activities, because in this stage is where the bulk of the formulation activities happen, and this is where you will get feedback, hopefully from your formulators, as to what particle properties they require. Again, you have scale-up activities, because necessarily your scale will be larger in most cases than in the preclinical and clinical trial phase 1 stage. And here you would also start looking at solid-liquid separation and drying of your product. It's important to get the process right at this stage, because you would freeze your process somewhere in the second half of your CT2 development phase, for the very simple reason that the API that you use in the formulation for the later CT2 trials has to be identical to the formulation and the material used in the CT3 phase. This doesn't mean that there is no work left for us crystallizers once you're in the clinical trial phase 3 stage, because this is when you start to work on activities towards commercialization. You finalize your scale up to the projected final scale of your process. carry out final process optimization within the constraints of the final process. Here you will do detailed QBD to understand your process limits and justify your process limits. And if it turns out that the initial process that you developed earlier on may not be viable for long-term supply, either for cost reasons or supply of raw materials, then you might start to initiate second-generation process development at this stage. Solid form screening that Stephen mentioned usually happens somewhere clinical trial phase 1, at the very latest clinical trial phase 2, and rarely in earlier development. We'll come back to that. And all of these crystallization process activities are accompanied by solid state characterization. This means understanding the solid state properties of your API and the powder that you generate during your crystallization process. Now, crystallization process isn't an isolated process that can be done without taking account of any other activities in process development. It is intimately linked with chemical process development, the synthesis development itself, with analytical development, and of course through scale-up activities with manufacturing. Crystallization itself rests on 3 fundamental physicochemical pillars. These are the solubility of your material. The solubility determines the type of process that you will select. It determines the lower working range for your crystallization process, and it also determines the maximum yield that you can expect from your process. Your starting concentration may be somewhere up here. Your final concentration may be down here, and the difference between these 2 concentrations is the maximum yield you can expect from your process. Nucleation kinetics and growth kinetics are the 2 other important factors. Both of these determine your particle attributes, your powder properties, your particle size distribution. The nucleation kinetics also provides the upper limit for your working zone for crystallization. Unlike solubility, nucleation is not a thermodynamic property of the system, it's kinetics, and nucleation is stochastic. This is why all these points here look rather random. It is a random process, but it's important to understand where nucleation occurs in order to control it for your process, in particular if you need specific particle size distributions. And the particle attributes will, as Stephen already mentioned, have an impact on your downstream operations, the solid-liquid separation, drying. It will have an impact on flowability of your material, and therefore it will also have an impact on further processing. Now I'd like to talk a little bit about early development activities. Now, we typically offer 4 standardized packages for early process development. And these standardized packages are simply a basic characterization of your material. So this is collecting physical data on your API, melting temperature, DSC looking for phase transitions, TGA looking at stability, an X-ray diffraction pattern as a benchmark for characterization later on, checking whether you have a solvate or a hydrate, and so on. And then we have 2 further packages that are screening packages. One is a polymorph screening package, one is a salt screening package, and the third is a crystallization process development package. And this is intended for the first crystallization process, should you not have one. So if you're relying on chromatography, for example, and you're looking to replace that with a crystallization process to reduce costs and increase efficiency of your separation process. Now, while these are standard packages, there is a workflow behind these that is more or less standardized, but every molecule is different. So the exact details of the activities behind each of these packages, in particular the crystallization process development, will very much depend on what you need and what your molecule needs. Let me talk a little bit more about solid form screening. So Stephen also already mentioned some reasons for doing solid form screening earlier on in the presentation. First and foremost, solid form screening is there to de-risk your process development. In an ideal world, you would like to have the most stable polymorph for your formulation, for the very simple reason that if you have the most stable polymorph, there is no risk of this form converting to something more stable with less favorable properties that may not have the bioavailability that you need for your drug product. However, polymorphs are not predictable, so you have to do experimental work, and you have to compare the forms that you find to ensure that you identify the most stable of those that you find, and then you still don't have a guarantee that you have the most stable form, but a good chance that you will have found the most stable form. Understanding the solid form landscape has other benefits. For example, interconversion of forms with potential to affect process development. It is not uncommon that a different form than the desired form is generated first during a crystallization process. So you generate a metastable form. This then transforms to the more stable form, the one you're looking for. And being able to control that, understand that is important. And you'll get a first indication of relationships between forms from a good solid form screen. Identifying solvates is important because solvates are generally a risk, both to process development, they may not dry easily, and to formulation. Solvates are usually not stable. There are very few APIs that are formulated as hydrates, for example, and most are anhydrous phases. And there is a good reason for that, namely their stability. And knowledge from screening can create efficiency in later process development. Stephen touched on this earlier. It may well be the case that your target solid form is difficult to purify. There are various reasons why this can happen, and understanding which other forms exist may help you identify one that can be purified, and you might have to introduce an additional step into your process, but with the knowledge gained from your solid form screening, you have a better chance of finding workarounds for difficult situations. And as Stephen also mentioned, solid form screening can be used for patent protection, and a good patent strategy based on knowledge of your solid forms helps to optimize your IP protection and also reduce the risk of challenges from competitors. And having the right form also helps to optimize and possibly enhance bioavailability. The most stable solid form may not be suitable due to poor bioavailability, and then you might start looking for salts, for example. You may be looking for co-crystals, and as a rule of thumb, different polymorphs usually have solubility differences in a range of about factor 10 with comparison to the most stable polymorphs. You can increase the solubility of your API using a salt by at most a factor of 100. And for co-crystals, a number of factor greater 1,000 is often mentioned. Whether this is accurate or not is a different matter. But this gives you an idea of how you can make use of solid form screening to identify a better form for your molecule. So what is a typical workflow for a solid form screen? Initially, solvent selection, we're assuming we're starting from zero knowledge. We get a powder which we assume is a particular molecule. We characterize the molecule. We make sure that this molecule is what it's supposed to be. And then we start looking at solvents, either through a qualitative solubility evaluation or quantitative two-point solubility. Usually we use both approaches, that we scan a range of solvents and then focus on those that are most suitable for screening activities and get more quantitative data. Then we carry out high throughput crystallization experiments, typically in neat solvents and solvent blends, and the experiments will encompass fast and slow cooling experiments and evaporative crystallization. At the scale we work at, we can't do true anti-solvent crystallizations where we have the API in a good solvent and we add a poor solvent, but this is what our solvent blends are there for. We approximate anti-solvent crystallization by using different solvent blends of good and poor solvents. Once we've done the initial crystallization experiments, which we do on these well plates here, so we can do up to 96 experiments in one go. We need to characterize and figure out what we've made. Initial characterization is by X-ray diffraction and Raman spectroscopy. This will tell us whether we have different crystal modifications, different crystal forms, whether we've generated something crystalline or something amorphous, or whether they're identical. And with the large amount of data that we're generating, we use cluster analysis to look for similarity in the diffraction patterns, group them, and then we will pick a certain number of promising experiments for scale-up experiments. Scale-up experiments are typically done at the 2 to 10 milliliter scale. They confirm reproducibility of the crystallization conditions. Polymorphism is notorious for being scale dependent. The smaller your crystallization system is, the more likely you are to find more polymorphs. Now, in actual fact, in most cases, we're not interested in many polymorphs. We're interested in process-relevant polymorphs, and this scale-up is the first test. Whether the polymorphs we found in the small-scale screen are actually relevant, whether they can be reproduced. And this is a simple matter of subjecting the powders to X-ray powder diffraction, making sure they're the same as in the first experiments. And then we carry out further characterization because we need to know whether we've generated polymorphs, whether we've generated solvates, whether we've generated a salt if we're making a salt. And XRPD cannot tell us whether we have done that. It just tells us whether we have the same or different modifications. And we use, for example, thermogravimetric analysis coupled with mass spectrometry to identify solvates. Solvates will become unstable at some temperature, so they will release the solvent. We can identify which solvent has been used with released with the MS. And from the mass differences, starting material and endpoint after mass loss, we can deduce the stoichiometry of any solvates. DSC to look for phase transitions, microscopy. This will tell us about polymorph solvates. For salts, we use NMR spectroscopy in order to identify salts. And then following the characterization, we determine the relative stability of the true polymorphs. And we don't consider the solvates because these are irrelevant at this stage. We want to know the relationship between the polymorphs. There is a subtle difference between polymorph screening and salt screening in that salt screening, first of all, requires a two-component system for crystallization, your API and the counter ion. How do we choose the counter ions? This is done based on pKa values of the API, and we calculate these in silico unless the pKa value is already known. And then we select suitable counter ions based on a general rule that says salts are formed if the difference in pKa between the two components is greater than 2. Sometimes you will see a number greater than 3 here. There is a bit of a gray area between difference of 0 and difference of 3, because there you might be forming something between a salt and a co-crystal. So how do we actually carry out these activities? So our high throughput screening is carried out in a crystallization robot, which you see here on the right-hand side of the screen. This allows us to dose solids and liquids, our solvents, automatically. We can prepare solubility measurements on this machine. The solubility is actually measured via HPLC. And we can carry out our crystallization experiments. And once we've carried out the crystallization experiments, as I mentioned, we use XRPD, top right here, for initial characterization, and Raman spectroscopy for spectroscopic characterization. Both of these are high throughput instruments, highly automated, and we can just run through all the successful experiments without great manual intervention. Our scale-up activities are then carried out after the cluster analysis. The cluster analysis, as I said, is simply a comparison of similarity of diffraction patterns and Raman spectra. This will give us a grouping according to predefined criteria. What you see here is one example of a cluster analysis, and everything that's in purple has been deemed to be similar. There is a threshold for similarity here. Every experiment that connects above this threshold is deemed dissimilar. Everything that connects underneath this threshold is deemed similar. We pick out the best possible conditions with a view to further process development for scale-up, carry out our scale-up experiments. The exact scale will depend on the properties of the material, confirm reducibility, so we go back to the XRPD, and then we have our further characterization, of which I've already mentioned, the thermal analysis, optical microscopy, NMR. We can also offer sorption analysis. This may be important for analyzing stability of salts, making sure that they are not hygroscopic, which again is a risk to formulation and process development. In some cases, it may be interesting to look at the true density of your solids. We have gas pygnometry for this. Density can also be used as a measure of relative stability. So in cases where there is doubt, this may be useful. And we can also look at particle size distribution, though this is normally not part of our screening activities. It's more something that we would use for crystallization process development. And then the final step, determining the relative stability of our polymorphs, we use competitive slurrying. So here you see an example of outcomes from a typical polymorph screen. This is a sample compound that we've used for demonstration processes. The compound is commonly known as ROY, red, orange, and yellow, and it's pretty obvious why it carries that name. Different polymorphs have different colors. They can be identified in some cases simply by looking at them. And you can see here microscope images from different crystallization conditions, different solvents, cooling crystallization, slow cooling, fast cooling, and different solvents. And these give you different outcomes. And characterization by X-ray diffraction shows you unambiguously that this yellow material here is not the same as this red material here. The relative stability is determined by competitive slurrying. And what is competitive slurrying? There are different ways of carrying out slurrying experiments, but the competitive slurrying is always a slurry of two solids, each in saturated solutions that are stirred together, typically pairwise, but you can do competitive slurrying with 3 or more forms if you want to. And over the time, the system transforms to the most stable modification due to solubility differences. Why is this? The most stable form has the lowest solubility, as indicated in the diagram here. The less stable form has a higher solubility. The concentration in your initial solution will be determined by the less stable phase, so it will be up here. But that means that the more stable phase is supersaturated and will start to grow. This means a reduction in concentration in solution, and in turn, the less stable phase starts to dissolve to restore equilibrium, and ultimately, the less stable form disappears, leaving only the most stable form. This is the only reliable experimental method for determining relative stability of polymorphs. Slurries of a single modification only tell you the stability of that modification over the time of the experiment, as there is no guarantee that a transformation will be observed if your phase is thermodynamically metastable. So finally, let's talk a little bit about competitive slurrying. Competitive slurrying is one method of determining relative stability of polymorphs, and it is the only experimental method without going into deep thermodynamics that easily allows you to distinguish the relative stability of polymorphs. How is it carried out? It is typically an experiment where you slurry at least 2 polymorphs, so it's typically carried out pairwise, but there's nothing to stop you from slurrying several polymorphs. And you take saturated slurries of both polymorphs, you mix them, you stir them for a period of time, and at the end, only the most stable polymorph remains. Why is this? Well, in the mixed slurry, the concentration of the solution will be dominated by the less stable modification. Less stable modifications always have higher solubility than the more stable modifications. So this is down here. And this means that the solution is supersaturated with respect to the more stable modification, and the crystals start to grow. At the same time, if the crystals of the more stable modification grow, they reduce the concentration in solution, and as a result, the less stable phase responds by dissolving to restore equilibrium. And ultimately, after a period of time, only the most stable form is left. A single slurry experiment of a given polymorph only tells you about the stability of that form over the time of the experiment. There is no guarantee that you will see a transformation to a more stable form if it exists. So at the end of a good competitive slurrying experiment, you should have evidence as to which form is the most stable. And this is an example taken from the Roy screening test. Here we've done a 4-component competitive slurrying, 2-component competitive slurrying. This was suspected to be a mixture of forms from a single crystallization experiment. And this is a single form slurry. And as you can see, this shows you the end point of the slurrying. All of these powder diffraction patterns are identical to the yellow form. And with that, the yellow form is the most stable of all the other modifications that were generated here. So you can be lucky, looking at the 4th example here, that you do see a transformation slurrying a single modification, but there is no guarantee. So I'd like to move on to crystallization process development now. Every molecule has its own story. So there is no one-size-fits-all single recipe for crystallization process development. But there are certain activities that have to be carried out. Once you've decided on the modification that you want to crystallize, you would typically evaluate solubilities, and in greater detail than you do during a solid form screen. You need to look at different crystallization processes, the parameters that you can modify, temperature ramps, anti-solvent additions, solvent volumes, anti-solvent addition rates, and the scale at which you do your development very much depends on which stage of development you are in. So in early development, you might be working at small scale. So here you see a multiple reactor for 16 parallel experiments. This is mainly used for solubility measurements, but can also be used for process development. We have its larger sister here with 8 parallel reactors and slightly larger scale of up to 10 milliliters. And here is where we have our first foray into process analytical technologies, because you want to understand what's going on in your crystallization process. And ideally, you want to look into the process as it is happening. This reactor comes with cameras for every single individual reactor slot, so we can observe how the crystals evolve over the crystallization process. Other techniques for in-process monitoring are focused beam reflectance measurement. This gives you a chord length distribution. This is somewhat similar to a size distribution of the particles, and you can see how the sizes change, how they evolve. You could possibly catch aggregation events, de-aggregation events, and even phase transitions in your process. Very useful here to have are also cameras, inline video microscopy to observe particles, how they form, how the shape evolves, what shape they actually have. And in-process infrared spectroscopy. This doesn't directly look at the particles, but you can monitor concentration changes and with that gain insight into your growth rate, which of course for a batch process is important for understanding the duration of your process, making sure you get the most out of the system. And crystallization process development doesn't stop with the end of the crystallization process. We want to get our particles out of our slurry. We want to dry them, and we want to do that as effectively as possible and without changing the properties of the material if we've designed our process to deliver a particular particle size. So that means we also need to look at solid-liquid separation, such as filtration. We need to look at washing. Impurities typically remain at the interface between the crystals and the mother liquor. They need to be washed away from the adhering mother liquor in order to reach the purity profile that is required. And drying, there are various technologies available for doing this as well. Okay, so finally, I'd like to present briefly two case studies. The very first one actually concerns a case where unexpectedly a more stable polymorph emerged during lab development in preparation for a manufacturing campaign. Now, for this particular API, polymorph screen had been carried out and did not detect this more stable polymorph. Unfortunately, the solubility of this polymorph was indeed a factor of 10 lower than the target polymorph. What are the consequences? Now, first and foremost, you have a loss of process efficiency. You need either considerably more batches, which means slower turnaround, which probably will mean a yield loss, or you have to look at other options. What are these options? One option may be to run with the existing process and keep your fingers crossed. Remember, I said that the more stable polymorph emerged during lab development. This doesn't mean that this will happen during manufacturing, but the risk is there. So you could use your plant as planned. You could hope that the new polymorph doesn't appear during manufacturing, but in any case, you should have a backup plan what you are going to do should things go wrong. Another option is to change the plant design, but very often that will not be possible because that means time and planning changes. There will be constraints due to use of the plant equipment by other projects, and ultimately changing the plant may lead to disruption of other projects, which is highly undesirable. Or you completely change your process. What options do you have there? You may look at different solvents. In this case, there weren't any good alternative solvents here due to solubility issues, but there may be cost issues, there may be processing issues. Or, forgot to mention this at the start, you could try to generate a different solid form, and in this particular case, we are converting an API from its free base to a salt. So you can try and convert to the salt before you carry out a clear filtration. The final API crystallization has to have a clear filtration to avoid particulates, and try to use the higher solubility of the salt to counter the lower solubility of the new form. Now, this also carries risks because you might find unwanted nucleation on the filter. So such a strategy requires lab studies to understand the nucleation behavior, quantify your risk, understand it, mitigate it. You might have a risk of unwanted byproduct formation. You might have solvent-salt reactions. This depends on the details of your individual process. At the end of the day, in this particular process, we actually ran with the first option, and we did have a backup plan for the case that the wrong polymorph was formed in manufacturing. We were very lucky. A number of batches worked as planned. And in the very final batch, we did find the new polymorph of the final intermediate, and the backup plan kicked into action. And that was simply a portion-wise dissolution and clear filtration, followed by solvent removal. And then everything carried on as initially planned. This is not something you want to happen, but unfortunately, there is no way of protecting us this for reasons I mentioned earlier. You can never predict the most stable polymorph. You can merely risk mitigate. The second example I'd like to discuss is a filtration example, so not directly crystallization. Slow filtration is quite frequently due to your particle properties. Typically, you have highly anisotropic particles, needles, fibers, or plates. You can see here two examples here. We have fibers on this side, we have plates on this side. They are quite frequently difficult to filter. You might have a compressible cake. This is often an accompanying factor with needle and fiber-like particles, or if you have very loose aggregates in your product, or you might be generating particle attrition by stirring during your filtration. And this is something that is not uncommon on a filter mixture. And this can lead to the generation of small particles on your filter, clogging of the filter. And if you have holdup, which you inevitably have in the manufacturing plant, the next batch might filter very, very poorly. So what are the remedies here? You can do different things. You can try and go back to the crystallization process and see if you can manipulate your particle habit. And this can be done in several different ways. The simplest way, if available, is a solvent switch. There are cases where a switch of solvent can lead to very different crystal shape without changing the crystal structure, the solid form that you're generating. In first approximation, the crystal habit depends only upon the crystal structure, but the theories neglect that the growth environment, of course, can play a role, and in solution very often does. And there is one example here where we had to carry out a solvent switch to change properties of the product. And you can see solvent 1 generates highly anisotropic particles. These don't classify as needles yet, but they are quite fragile and they lead to particle attrition during filtration. And solvent 2 yields to more isometric, very compact particles that filter very well. Another popular approach for manipulating particle shape is temperature cycling. And temperature cycling is based on the fact that there is no single crystal growth rate. You can look at an average crystal growth rate, but the fact of the matter is every distinct crystal face has its own growth rate, and its temperature dependence of the growth rate will be different. And you may be able to exploit this by partial dissolution of the particles and then recrystallization with a sinusoidal temperature cycle in order to manipulate the shape of the particles. Sometimes the effect can be quite striking, sometimes it's subtle. I would call the effect here for this example subtle. You can see very thin needle-like particles in the left-hand picture. In the right-hand picture, they are more compact, but they're still fairly anisotropic. But in most cases, this will not work. And again, there is no way of predicting whether this will work. A few experiments should give you a very quick insight whether this is a good strategy or not. And in most cases, a much better strategy is to choose appropriate manufacturing equipment based upon filtration studies. What does this mean? So typically, filtration studies aim to determine the filter cake resistance, quantify the filter cake resistance, and then make decisions for scale up of your filtration process based on the properties of your filter cake. And the filter cake resistance isn't the only quantity that is important. Compressibility is also an important issue, and compressibility of a cake can be found by carrying out filtration experiments at different pressures. And if the cake resistance changes with pressure, then you are dealing with a compressible cake. And depending on the magnitude of the compressibility, this may be a severe problem, it may not be a problem at all. In this particular study, the material was almost fiber-like, and we looked at filtration properties. This is our filtration equipment. It's a pressure filter. The filter actually sits underneath the vessel here. We fill our slurry into here. There's a bayonet that attaches the filter. We can use appropriate filter media that we use in manufacturing, and we measure the we acquire the filtrate, and we measure the evolution of filtrate volume with time, and from that we can calculate the filter cake resistance. We support these activities with simulations. We do experiments at 3 different pressures, and in this particular case, we found a filter cake resistance of 1.5 to 10 to the 11 meters per kilogram. And if we look at this table here as a guide, this tells us that this is a slow filtering material, and the best equipment for this type of material is a belt filter or a centrifuge or a pressure filter. Pressure filters are not very common in the pharmaceutical industry. We have centrifuges and a belt filter. But since this material is compressible as well, and centrifuges are a poor choice for compressible materials, we opted for a belt filter. You can see an example of a belt filter here. Why do we use a belt filter in such cases? The cake that we generate on the belt filter is very thin. And any issues due to compressibility of the cake, due to high thickness of the filter cake, tend to be mitigated by the thin layer of cake on the belt filter. Typically, we're talking about 5 centimeters maximum on a typical belt filter. And with this, you can rapidly increase your processing time. We have had examples where we've increased processing times from 3 weeks on a filter nurture down to 3 days using a belt filter. Now, this is may still seem a poor comparison, but a large fraction of those 3 days were actually subsequent washes of the material off the belt filter and not the filtration on the belt itself. That was considerably faster. And with that, I come to the end, and I'd like to summarize the presentation. So, in summary, crystallization process development is an integral part of API synthesis development throughout the development life cycle. A good intermediate crystallization strategy has significant regulatory benefits, as Stephen outlined. In early development, early development and in particular form screening provides valuable data for efficient late-stage development. And it is crucial to include crystallization in your process development to ensure a cost-effective and robust process suitable for filing. And failure to pay sufficient attention to solid-state characterization and crystallization can lead to nasty surprises at the most inopportune time and lead to unnecessary delays and put patients at risk. Thank you for your attention.
And thank you for that great presentation. A few of you have already submitted questions, so we're going to jump right in. The first question is, when should one start with solid state characterization, crystallization process development? Who wants to take that?
This is not an easy question to answer. Yeah? So you should start as early as possible, and in most cases you will have some indication from medicinal chemistry whether your material crystallizes or not. You will have a process that more likely than not is unsuitable for manufacturing. So you should start developing an initial process probably at the stage where you're thinking about your tox campaign. No. This will be fairly low intensity, good enough. You won't put a lot of effort into it. And it may well be that you've completed your activities in just 2 to 4 weeks. You might opt to start a little bit later, run with an existing process, because you think it might work. You may have to change tack at a later stage. You might need to change solvents if they're unsuitable for large-scale manufacturing, or issues turn up that you haven't anticipated.
And I'd like to add, maybe, there's always a trade-off between risk of failure. Many phase 1 compounds don't make it to phase 2. So how much do you invest early on, as opposed to the more that you invest early on, you are able to get the benefits of those improvements in subsequent campaigns and have a better control. And so it's really a discussion. I would say talk to your partner, your manufacturing partner, understand what the cost benefit analysis is of doing each stage of the crystallization, and then jointly decide what the best process is to move forward.
Thank you. Second question we have is, what is the best timing for a solid form screen?
Again, there is no clear-cut answer to this. In the past, because of the risk of failure of early development compounds, many companies have opted to do this as late as possible during clinical trial 2 development. But we've seen a shift in the strategy, and more companies are opting to do solid form screening either at the CT1 stage or even during preclinical development. Of course, you're increasing the risk of failure the earlier you go, and it may be a valid strategy to do a limited solid form screen as early as possible, so not put too much effort into it, and do a comprehensive form screen later on in your development. But you shouldn't really leave it too late, because whatever you find there may have a significant and potentially detrimental impact on all the rest of your synthesis and formulation development.
Thank you. Third question, another simple one. How long does crystallization process development take?
So this very much depends on what stage of development you're at and the requirements of your molecule. And as I said earlier in the presentation, every molecule is different. Every molecule has its own story. If we're talking about early phase development, you would typically be looking at maybe 4 weeks of development plus minus a couple of weeks for the very first process. For CT1 development, it can be anything between 6 weeks and 3 months of development, depending on how complex the system is and what your requirements as a customer are. The bulk of the work, as I mentioned, is done during CT2. And there you have to probably plan for at least 3 to 6 months development work. Again, very much dependent on the complexity of the system, how much effort you're willing to put in at that stage. And I have seen projects close to commercialization in CT3 where several man-years of effort have been put in to get everything right, to fully understand the process, to understand the process ranges, mode of failure. It can be considerably less. So don't let that put you off. It very much depends on the molecule.
Right. Subsequent question, can you please share what the timelines are for different solid state chemistry activities?
So if we're talking about the screening, typically we plan for a maximum of 6 weeks from receipt of material to delivery of report for any form screening. We plan for the same amount of time for crystallization process development. The solid form screening can usually be done in somewhere between 4 to 6 weeks. Again, it depends on the material and what surprises we encounter. For a benign material, will be faster. For a more complicated material, it will take a bit more time. Crystallization process development usually fills out the 6 weeks. And the solid form characterization is usually done very, very quickly. We can do that in maybe 2 weeks, the full package.
Thank you. This one sort of follows up on the last, really. Can you please comment how much time could be saved if crystallization and solid state chemistry activities are done early enough, or how much time might be lost if they're done too late?
So it again very much depends on what the reason for any time losses is. If you find a new polymorph despite your best efforts, you might be losing a few months in redeveloping a crystallization process. At the same time, you might be losing considerably more time for reformulation, should that be necessary. It may well be, remember right at the start of my part, I mentioned everything is integrated and we very much depend on the synthesis development as well. Impurities can influence crystallization. Any changes to the synthesis can change the crystallization. So that can lead to changes and need for redevelopment. It's unpredictable. Sometimes the problems that occur are easily solved within a very short period of time. I have seen projects that were very difficult, where the effects were very subtle, very difficult to pinpoint, where the delays were months up to a year. There's another factor to consider, your planning. If you are working with custom manufacturers, you're working with several custom manufacturers, you're planning clinical trials, what happens if there is an unexpected surprise due to not doing the work early enough, and the API manufacturers don't get their material ready in time for the formulation, and the formulation is delayed and does not produce in time for your clinical trial? What happens to your time slot? It disappears. When are you going to get the next one? In worst case, you might be waiting 2 years for another clinical trial slot. These are the complexities and the risks in process development.
Yeah, and I think, maybe to summarize it up is ignore at your peril. Talk to your partner, your manufacturing partner, understand what the benefits and the risks are, and put together a plan together. I think that's really the message of this webinar, is that you really need to make sure that you include crystallization process development in every stage of your process. And clearly there's a cost benefit analysis, there's a risk analysis, and make sure it's part and parcel of the process that you're developing.
Thank you. I think that leads neatly on to the next one, which is, what are the advantages of having API development and subsequent formulation development services within one service provider? Will it save time in the whole process?
Yeah, absolutely. I think it's key, as Matthew explained, as you develop your API process, you may choose different process conditions, for example, for filterability, but that has an impact on the downstream processing for the drug product side. And so filterability can have the particle shape, the size, can have an impact on the how well the part behaves, the compressibility. And so whatever changes are made need to be communicated and discussed with the formulation team on the drug product side, and needs to be done hand in hand. As I expressed in my slides, that if you do not do that, that can cause you easily a 5-6 month delay with regards to having to redo formulations and redo studies on the drug product side.
Right, just 2 more questions. First, what's the difference between a polymorph and a salt screen?
The key difference is that you're looking for different solid forms of your API in a polymorph screen, where these solid forms are either chemically identical to your starting material. That means that they may have different crystal structure, but the content of the crystals is chemically the same. Or you're identifying solvates. So these are binary two-component crystals that consist of your API and a solvent, usually in a stoichiometrically defined composition. And for the salt screen, you're looking at whether you can generate a salt. One prerequisite there is, of course, that your API has to have an ionizable group, an amine or an acid group, and you have to be able to identify suitable counterions. The actual work that is done there is very, very similar, but the goal is different.
Thank you. And the last question, what are the benefits of a solid form screen?
The major benefit of a solid form screen is the wealth of information that comes out of such activities. You're doing a large number of crystallization experiments, looking at different conditions. You're looking at a large number of solvents, evaluating solubility. You will get information on crystallizable solid forms, whether they're polymorphs, solvates, or salts. If you're looking at co-crystals, different co-crystals, their stability ranges, and you can use this to focus and target your subsequent crystallization process development. And of course, the characterization that is done for these forms can help to focus and target your formulation development.
Thank you. That's all the questions.
Okay, well, thank you very much. We appreciate everybody's time and attention, and thank you for listening in.
And thank you from me too. Thanks for your attention.